Hello, my name is Leanne Shabana, and today I will be presenting a glow discharge optical emission spectroscopy analysis of silver nanoparticles on pristine plant tissue. The individuals who contributed to the research include Aldo Hernandez and Kevin Finch as part of the GDOES lab research group in the Department of Chemistry and Biochemistry. So let's begin. Nanoparticles have been shown to have applications in many different fields, such as the fields of medicine and agriculture. Our research involves the field of agriculture and deals with the study of plant nanoparticle interactions. Specifically, nanoparticles have been shown to either improve seed germination or disrupt nutrient uptake by plants. Nanoparticle plant interaction has been studied in the past by applying many different techniques that require extensive time to undertake and that also require the implementation of tissue encapsulation in their sample preparation. Glow discharge optical emission spectroscopy, however, allows for ultra-high throughput nanoparticle analysis and was used in our case to map pristine biological soft tissue. This is the first time that glow discharge optical emission spectroscopy elemental mapping has been applied to such biological tissue. As nanoparticles become increasingly exposed to the environment, quick and thorough analysis of these particles is required. Glow discharge optical emission spectroscopy involves the breakdown of argon gas in a chamber and the sputtering of the nanoparticle sample into a plasma, where ions are excited and emit a photon of wavelength characteristic of the sample. The project undertaken here will show how glow discharge optical emission spectroscopy was successfully used in the elemental mapping of pristine plant leaves. As I continue on to my GDOES elemental mapping section, I will explain the elements of our imaging system. To begin, our light source is a glow discharge lamp. Light first goes through the collection lens as pictured here, which collects it and also demagnifies our image. The light continues on through the collimating lens, which aims to make all the light beams parallel. Later on, the light goes through our 330 nanometer filter lens, which aims to selectively transmit only the light emitted by silver atoms at 330 nanometer. The light continues traveling until it hits the mirror, which reflects it at a 90-degree angle, into the focusing lens, which focuses it into the spectrograph. The point where the light meets is the focal point, and that's where an image is created. The 2D detector, which is the camera in our case, gives spectral information along the full Y dimension and a portion of the X dimension. As our mirror moves, the rest of the X dimension is filled in to give a full image of the nanoparticle spot. As we continue on, I will explain our method of sample preparation. The sample collected was a leaf from the Madagascar periwinkle plant. In an effort to dry it, the leaf was sandwiched in between two filter papers, which were then placed in between two glass slides. This setup was then left in the lab to dry under ambient conditions. Then, the sample under analysis was mounted onto a glass slide using nickel adhesive paint. Two other mounting protocols were undertaken and involved the use of Kapton tape or double-sided copper tape, as seen here in Figure 7. Finally, as seen by Figure 8, 20 microliters of a 40 nanometer nanoparticle suspension was deposited in the middle of the leaf that was mounted on the slide with nickel paint. Let's continue on to the results section. The top image here is of the plant leaf after a nanoparticle deposition. The image to the left of that is of the plasma formed in the glow discharge analysis of the plant leaf. A 2D elemental map of the silver emission can also be seen here. And finally, the 2D map overlaying the plasma formed is seen in the final image at the bottom. Our data is analyzed by calculating a limit of detection. The limit of detection is actually established through a background equivalent concentration. The background equivalent concentration takes into consideration the relative standard deviation of the background and a signal to background ratio in addition to the mass of the nanoparticles. Later on, the LOD can be calculated by dividing the background equivalent concentration by the nanoparticle deposit area in order to obtain an LOD of 35.4 picograms per millimeter squared. As I continue on, I will discuss our findings. The nickel paint mounting protocol proved to be the most effective. Our LOD was calculated to be 35.4 picograms per millimeter squared under non-optimized operating conditions. This means that glow discharge optical emission spectroscopy successfully allowed for the elemental mapping of silver on plant tissue. Elemental mapping was actually undertaken in 4 seconds, which is approximately 1,000 times faster than when a method such as laser ablation inductively coupled plasma mass spectrometry was used in other works. This means that many more samples can be studied, leading to a more comprehensive understanding of nanoparticle plant interaction when implementing glow discharge optical emission spectroscopy. Finally, our future work will aim to study the effectiveness of other leaf mounting protocols in an effort to improve our limit of detection. In addition, we will aim to optimize our elemental mapping operating conditions, also in an effort to improve LOD. Finally, we will aim to study plants grown under nanoparticle exposure in an effort to monitor cell uptake. Work on microarray substrates is currently being done as part of a nanoparticle cell uptake study. Aldo Hernandez's poster can be referenced for further information. Thank you so much for your time.